Wonderful. So again, we are very pleased to have Dr. Jamie Zeitzer here with us. He is the Associate Professor of Psychiatry and Behavioral Sciences and the co-director of the Center for Sleep and Circadian Sciences at Stanford University. He also holds a position as a health science specialist at the VA Palo Alto Medical Center. He has more than 25 years of research experience examining sleep and circadian rhythms in humans as well as animal models. His current research mainly focuses on the impact of light on human behavior, circadian-based sleep disorders, and the use of wearables to improve human health. Thank you so much, Dr. Zeitzer, for being here, and we really appreciate this, that you're spending your time with us. And I hand it over to you. Thank you so much.
Great, thank you so much, Susan. Let me, so I assume you can see my screen? All right, great. So if you've got any questions, any burning issues, digressions, I'm more than happy to chat. I sadly have the attention span of a fruit fly, and so I'm happy to go off on lots of tangents as you would like. So modern sleep medicine, or how I learned to stop worrying and love my sleep. So I'm going to be going over lots about sleep, about the basic biology, and about kind of how we understand wearables and what we understand about sleep and what to do to improve it. So let's get into it. So this is kind of the overall presentation structure. So, the first thing is what exactly is sleep? And there are various ways that we can define it. One of the ways we can define it is just behaviorally. And if you want to look at sleep in a variety of different organisms, sometimes you do need to kind of look at different behaviors. And so one of the fundamental things is, well, there's not a lot of movement. Now, I say there's not a lot of movement. There still is movement. Some people think that they don't move at all during sleep. And if you want, you can take a camera and set it up, take your phone and do a time-lapse recording of sleep. And you'll see that everyone moves, even a little, during sleep. So there's some movement, though not much. There's a stereotypic posture. So basically, animals, people included, usually sleep in beds. Animals sleep in different positions in different locations. There's reduced responsiveness to external stimuli. So what this means is that it's not that you're not responsive. You're still getting information into the brain. It's just that you've got reduced responsiveness. So you can still hear things, smell things, taste things, feel things. It's just that there's a higher threshold of salience before you respond to them. So I show a picture of a train here because it's one of the typical examples is you move in to a new apartment. It's near a railroad track, and the train comes by 3 a.m. and wakes you up, and does this for a few days, and then it stops waking you up. So your brain is still hearing the train at 3 a.m., but now it realizes that it's not an important piece of information, and so it blots it out. So it's just not responding to it. So it doesn't wake you up in response to that train anymore. Now, if the train's late, your brain can actually wake you up because it was expecting the train at 3 a.m. And when it doesn't come, that lack of information can actually be important. So this is all to say the brain is still functioning, it's still active, it's still processing information. It's just that what it considers important changes. Oh, and I should mention here, as people age, this ability kind of declines. And so as people get older, think of little kids, they don't wake up to anything, and then people in their 70s and 80s, there's a stray piece of light, in the room and people are waking up. There's a random sound, they're waking up. So your ability to kind of do this kind of declines with age. And this is one of the many reasons that sleep can be disrupted in older individuals. Fourth thing is that it's reversible. Just to kind of differentiate it from coma and death. And the fifth thing is that it's under homeostatic control. And this is one of the key aspects of sleep, which is that it is in homeostatic regulation, which basically means is that it's going towards an equilibrium or set point. So if you don't get enough sleep, you get more tired. As you sleep, you get less tired. So again, you can think of it as like an hourglass kind of mechanism. So I guess the first question here is, well, how tired are you? So this is a little scale. So if you just give yourself a score here. For each of the 8 questions, give yourself a score of 0 to 3 based on the chart here. So your end score should be somewhere between 0 and 24. And so this is not how tired you are right now at this moment, but just how tired you are in general. So I'll give you a second to get yourself a score. Okay, so hopefully everyone's got a score. Well, if you want to contextualize how tired you are in driving, this is about, I put the wrong scale. I'm so sorry. This is the Stanford Sleepiness Scale. I apologize. So basically, if you are scoring between a 0 and 8, you're okay. If you are scoring above an 8, that's the federal safety limit in terms of its equivalent function for your blood alcohol content. And if you are scoring above a 12, your driving performance would be similar as if you were legally intoxicated. So basically, if you're above an 8, that's not good. If you're above a 12, that's really bad. Yet people exist every day like this and kind of function. And one of the problems that we have in sleep and thinking about sleep, especially as it relates to sleepiness and daytime function, is that how you feel, how your subjective sleepiness is not necessarily related to objective measures of how functional the brain is working. And so if you take individuals who are constantly or commonly sleep deprived, so if you look at individuals working extended shifts in the hospital or overnight shifts in the hospital, individuals flying planes at night, long distance truckers, these are individuals who are constantly exposed to sleep loss. And in that kind of situation, you really have, one of 2 possibilities. One is you could just be, unhappy in being sleep deprived and tired all the time, or you can start to ignore the signals of sleepiness. Now, when you do that, and we and many others have looked at individuals who are like this, and you bring them into the laboratory, you bring someone like, you bring a surgeon into the laboratory, and you sleep deprive them, and you have them rate their sleepiness. They'll say they're not sleepy, and genuinely they do not feel sleepy. Yet you test them and you see that their performance drops off just like everyone else's. So basically their brain is functioning as if it were sleep deprived, yet they don't realize this. There's no subjective realization of how tired you are. And so there's this big disconnect between how tired you are and actually how your brain is functioning. And again, if you're in a position, say working at a hospital where there are patients all the time, and you are put into a position where you're constantly being sleep deprived, you often don't realize how tired you actually are. And lots of medical errors happen. And I don't know if you remember a couple of years ago, there was a plane that missed the runway at SFO. And these individuals, the pilots, it was Asiatic Air, and the pilots coming in were flying in at just the wrong time of day for their brains. And they were very tired and they misestimated the position of the runway. So we've got behavioral definitions, but since the last 70 or so years, we really have been measuring sleep using something called the polysomnograph. And in polysomnography, this is what we use to kind of really define actual sleep and wake states. We're using 3 main signals. We're using the electroencephalogram, which is measuring basically hundreds of millions of neurons firing at the same time, measuring the EOG or electrooculogram, which is measuring eye movements, and the EMG or electromyogram, which is measuring skeletal muscle tone. The EEG, for those who are interested, basically you've got when you're recording it on the scalp, it's around 100 microvolts. Basically, what you're looking at is you're looking at lots and lots of neurons and how they are firing together. So if you get bigger signals, that means you have more neurons firing together. If you have smaller signals, it means that they are not firing synchronously with one another. If you do record the EEG at the actual surface of the brain, it is a good 100-fold higher in terms of its amplitude. And you can do that in various kinds of brain surgeries. So how do we take this information and kind of think about sleep this way? Well, we can basically take these three different signals, the EEG, EMG, and EOG, and typically we will separate it out into three different states of consciousness: wake, non-REM, and REM. And REM stands for rapid eye movement sleep, and NREM stands for non-rapid eye movement sleep. And you can see here in this kind of chart that each one can be defined by different signal types. So in wake, and I'll show you examples of this, you've got a mixed EEG, you've got high muscle tone, you've got random eye movements. In non-REM, the EEG is synchronized, the muscle tone is low, similar to basically kind of quiet wakefulness, and again, random eye movements. And then in REM sleep, the EEG is similar to wake, it's fairly indistinguishable. The EMG is very low, so you actually have actively suppressed muscle tone. And the EOG indicates saccadic eye movements, or these are the eponymous rapid eye movements. So basically, you've got your eyes darting back and forth. And this is kind of what it looks like in terms of a signal. The EEG, this is what we would consider a mixed EEG. And the eye movements are these deflections. Eye movements, basically, you can pick up eye movements in this kind of signal. You put electrodes on the corners of the eye, and there's a 1 millivolt difference across the eye from the back of the eye, which is the retina, which is neural tissue, to the front of the eye, the cornea, which is non-neural tissue, and that movement in that kind of electrical field creates these kinds of deflections. This is non-REM. So if you take a note at the EEG signal here, this is EEG during wake, and this is EEG during non-REM. And so you can see the signals are much larger. These larger signals indicate that more neurons are firing synchronously. So not that more neurons are firing, but that just that they are firing synchronously. And when they fire synchronously, you get these larger signals. And then if you look at the EMG here, you'll see that the EMG during wake is considerably higher than that during non-REM. We can further break down non-REM into typically three different stages: stage 1, 2, and 3, or N1, N2, and N3, and sometimes N3 is called delta sleep or deep sleep. And you can see that the EEG, which is represented here, varies across these stages. So in deep sleep, you've got these very large waveforms that we call delta waves. It's between 0.5 and 4 hertz in terms of frequency. This is what people think of as kind of the deepest sleep. Stage 1 is very similar to wake. It's a very kind of liminal state. It's not a very deep state of sleep at all. In fact, you can have situations where parts of your brain are in stage 1 sleep and other parts of your brain are awake. This is more common when you are sleep deprived. If you're sleep deprived, you can have different parts of the brain in different stages of sleep. And in fact, one thing I'll talk about a little later is that in individuals with insomnia, this is a very common occurrence. When you're trying to fall asleep, part of the brain will fall asleep and another part will be awake. So this gets to something which is kind of a newer concept in sleep, which is this idea of local sleep, which is that when you think about sleep, it's not that the whole brain is awake or the whole brain is asleep. If you're a good sleeper, that's exactly what's happening. But if you're not a good sleeper, or if you're awake and you're sleep deprived, what can happen is you can have pockets of the brain doing different things. So you can have this part of the brain being awake and the rest of it being asleep. And then this part falls asleep, and then this part wakes up. And then this part wakes up. And so you can have different kinds of situations where different parts of the brain are doing different things. And if you're trying to fall asleep, this can be not a great experience. You can basically feel like that you're neither really awake nor asleep. And if you're trying to be awake, it's again the same kind of thing where you don't have the situation where you're really awake or really asleep. And you can have lots of problems in terms of lapses of attention. You can fall asleep while driving. You can fall asleep while working. It's not a great situation. So these are the EEG signals that you get from different parts of the PSG. We then go to REM sleep, and in REM sleep, the EEG looks very similar to what you get during wakefulness. And then you get these sharp deflections in the EOG, and these sharp deflections represent the eyes moving back and forth or darting back and forth. One last layer of complexity for this, you have, this is called the hypnogram. It's how we represent changes in sleep stages across the night. So you start out awake, and then you go into N1 sleep through N2 and N3, and then you go into come back into N2, N1, and then REM sleep, and that you kind of cycle through these non-REM and REM cycles throughout the night. And when people talk about a 90-minute cycle, they're really referring to a 90-minute non-REM plus REM cycle. So if you add in non-REM plus the REM, that's about 90 minutes in most individuals. And this cycle length is fairly constant across the night, but as the night gets longer, you get more REM sleep. So you have more REM sleep at the end of the night, less in the beginning of the night. And then you have more deep sleep in the beginning of the night, and more N3 sleep in the beginning of the night, and less at the end of the night. REM sleep is often thought of as dream sleep. Dreaming probably occurs during all stages of sleep, but your recall or your memory of dreams is definitely the highest when you are awoken out of REM sleep. Following on this line, we can also look at how sleep changes across the age. Notice this is not a linear scale here, but newborns spend about 16 hours asleep. Unfortunately for most parents, that's not a continuous 16 hours. It's highly fragmented, but 16 out of the 24 hours, they're often asleep. And this very rapidly drops off to 14 hours at around 3 months. And then you can see that it kind of declines over the course of a lifetime. Here into the adult range, you get around 8 hours of sleep, and this drops to around 6 hours of sleep in a 90-year-old. In the beginning, newborns, they spend about half their time in REM sleep and about half the time in non-REM sleep. But this very rapidly goes to about 20% of your time in REM sleep and 80% of your sleep time in non-REM sleep. And that ratio is fairly constant starting at about 2 years of age. So this gets into something I'm just going to talk about briefly, which is, well, we all sleep and we obviously need to sleep, but what is sleep doing? What is the function of sleep? Now, I'm not a huge fan of this question. Most people don't go around asking what the function of wake is, but we do ask this question of what the function of sleep is. And there are various things that happen during sleep and appear to be optimized during sleep. One is this issue of endothermic debt, which is that as endotherms, so we maintain our body temperature, this is metabolically very costly. If you're sleeping at night, your body temp drops around 1 degree centigrade. While it doesn't seem like that much, that helps save quite a bit of energy. And so it's possible that, that 1 degree drop during sleep is important for saving energy. So that might be a function of sleep. Memory consolidation is another thing that has been proposed to happen during sleep. And so you can think of memory consolidation as kind of the movement of information from short-term memory to kind of long-term knowledge. So you can memorize facts during the day, but these facts then will get integrated into what you already know during sleep. And so that's kind of one of the things that's happening during sleep, is this form of memory consolidation. And then another one is metabolic homeostasis, which is basically you spend all day long, your brain is doing stuff. And all that stuff costs energy and it produces toxins. So one of the things that's happening during sleep is that you're regaining your energy, you're moving energy around, you're gaining new energy stores, you're getting the toxins out of the brain. That's this glymphatic system, is something that was discovered only a few years ago, which it seems to be. So glymphatic is a combination of lymphatic and glia, and it appears to be a way for the brain to remove toxins, and flow through the glymphatic system is enhanced during sleep. So basically, one of the things that might happen during sleep is all the consequences of kind of all this thinking and doing stuff during the day, and production of all these toxins and use of all this energy. There's kind of a stabilization of this during sleep and renormalization. So that's possible. And it's also possible is we don't know yet. And there may be other functions that are happening during sleep. And so really, this is an area of great interest and a lot of activity. But it's something that, we spend a third of our lives doing, and we're not really sure why we have to. But if we don't, we know that we do suffer the consequences during the day. But if you take a look at across different species, that's not always the case. You have some species that can flip a molecular switch. So there's the white-crowned sparrow that during non-migration season, sleeps a normal amount at night. And if you keep them up at night, it'll perform poorly on a cognitive test. So they basically act sleep deprived. But during migration season, they only sleep a couple of minutes a day. And there doesn't seem to be any negative impact on its cognitive function. How it does that, we don't understand. But a lot of people would like to be able to take that pill where they could switch their brain to not needing any more sleep. So before I go on to kind of what is happening during sleep, are there any questions so far? No?
There is none in the chat, but please, everyone, if you have any question, feel free to unmute and ask your questions, or put it in the chats and I will monitor the chat.
Okay, so physiologic manifestations. So basically what's happening during sleep. So, when you look at the body, lots of things are changing during sleep. But as I mentioned, there are thermoregulatory changes during sleep. There's a degree centigrade drop in brain temperature and body temperature during sleep. And this is due to a change in hypothalamic set point. So in the hypothalamus, there's basically a thermostat that's determining what your body temperature should be at. That temperature is set to a lower point, which causes your brain temp to drop, your core temp to drop. And in order to actively drop your brain and core temperature, you need to release the heat somehow. And you do this heat release through something called anastomoses. And anastomoses these are arteriovenous anastomoses. These are places where arterioles and venules merge without a capillary bed in between them. And these are notably happening in the non-hairy parts of the body, the palms of your hand, your forehead, and the soles of your feet. And this is where you do most of your active heat loss. And so if you're trying to lower your brain temperature, you're going to increase heat loss here. And so you're going to have an increase of temperature in the hands. And this is one of the things that happens right before you fall asleep, is you see these temperature shifts in the skin, and you see lots of active heat loss in the hands. During REM sleep, there's no active heat regulation. So during REM sleep, you don't have the ability to control at a central level what's happening in the periphery. And this is because during REM sleep, you have active suppression of all motor neurons. So anything that is happening through the spinal cord is being actively suppressed during REM sleep. You do not have inputs and outputs to the brain via the spinal cord during REM sleep, which means that if it gets very hot in a room, you have no ability to cool off centrally. So your brain can't detect it and coordinate the body to cool off. And if you remember, infants in the first couple of weeks are spending 8 hours a day in REM sleep, which means 8 hours a day they're unable to thermoregulate. And for anyone who's had an infant or worked with infants, this is one of the key reasons that you have to basically thermoregulate for them, is because for 8 hours a day, they can't do it. And the ratio of their body mass to surface area is such that they can very, very rapidly lose and gain heat. And so it's one of the reasons that people are always, and for good reason, warning people about leaving infants in cars during the summertime, because again, they can overheat extremely rapidly, especially when they are in REM sleep. They're changing the cardiovascular system. These are supportive of heat loss. There's decreased blood pressure and cardiac output, as well as increased vasodilation during non-REM sleep. During REM sleep, there is an increase in blood pressure variability, which is secondary to changes in vagal tone, as well as decreased cardiac output. But then there's a slightly different aspect of vasoconstriction and dilation. So you have increased constriction in striated muscle, but then all other muscle has an increased vasodilation during REM sleep. During the autonomic system, during non-REM sleep, there's nothing really, well, there are changes, but there's nothing really consistent. But during REM sleep, there's increased tone in both parasympathetic and sympathetic systems. Renal activity changes similarly in both non-REM and REM sleep. And it's basically a coordinated way to lower urine volume and have more concentrated urine, which again, kind of makes sense. You don't want to have to urinate as much at night. Same thing with the stomach. You've got less stomach digestion. You've got decreased gastric acid secretion and decreased swallowing and esophageal motility. The muscle tone is decreased during non-REM sleep, again, similar to kind of relaxed wake. And in REM sleep, you have active inhibition. So this is when people say, during REM sleep, you're kind of paralyzed. This is true. You're unable to move. Your cranial nerves are not affected. This is just spinal motor activity. And it's such that, again, you have no central control over peripheral musculature. Sometimes people will say, well, this is so that you don't act out your dreams. We don't know if that's why, but it definitely is a benefit that's happening. In infants, it actually serves a slightly different role. One of the things that you can see in infants, if you've got an infant or had an infant, remember them when they're sleeping, you see all these little twitches. These twitches are happening during REM sleep, and this is the brain basically learning how to control different groups of muscles in the body. So basically, you've got a situation where everything is suppressed. You've got all suppression of muscle tone or spinal motor activity, and then you get a little breakthrough. So you get like one neuron that fires, and then that triggers a muscle twitch, which then feeds back into the brain. And this is kind of a way that the brain can learn how specific neurons in the brain are connected to specific muscles in the periphery. When you look at respiration, there are changes there as well. During non-REM sleep, there's a decrease in ventilation. There's increased CO2 levels and decreased oxygen levels, increased upper airway resistance, and decreased responsivity to bronchial irritation. One of the reasons that if you've got a cold or you've got an irritation in your throat, you can sleep through it. It doesn't always wake you up. During REM sleep, your breathing becomes much more irregular. Again, you have this increased upper airway resistance and decreased responsivity. And there's no chemical feedback through the spinal cord. It's only mechanical feedback, which is local. So these respiratory changes. You also have changes in endocrine function. So here are a couple of plots that I'm going to show you of endocrine function. In these plots, kind of this purple area is representing the timing of sleep. The black line here represents what happens to this hormone during sleep. And the gray line is what happens during this hormone if you are kept up during the night and not allowed to sleep. There are lots of other controls to make sure that other things aren't influencing it. But this is one example. This is thyrotropin, or TSH, which controls the release of T3 and T4. It's involved in controlling global metabolic rate. And so what you can see is that normally what's happening is that you get an increase of TSH in the evening, and then at night it drops off, and it's kind of lower and getting lower throughout the night. But if you stay awake, it keeps on rising and getting higher. And so this is one of the things that we think is involved in driving abnormal hunger during night shifts. So basically what's happening is that you're up at night, and your TSH is getting way too high, and it's telling your body you need food. And the amount of food that you need is going to, this is a very gross way of controlling metabolic needs. And this is basically overshooting your metabolic needs. So you're going to overeat, and you're also going to be going for things that have that are high in calories, high in sugar, high in fat. And this is probably one of the culprits of driving these kind of abnormal hunger signals when you're working at night. If you look at cortisol, there really isn't much of a change whether you're sleeping or not. And the reason is that cortisol really isn't so much affected by sleep, but it's instead affected by something called the circadian clock. And this circadian clock, it's a 24-hour clock, and it kind of basically controls the timing of, well, most everything in the body and the brain. It's just a question of the magnitude of that control. And it allows for anticipatory changes. And so in this case, you don't have much of a difference between sleep and wake. But what's happening is that cortisol is rising in anticipation of when you're supposed to wake up. And one of the things that cortisol is doing is that it is mobilizing your energy, predictive of when you're supposed to be waking up. And this is super important because it gives you a lot of active energy. If you wake up at the right time, you can get up and you feel good and you've got lots of energy to do it. Metabolically speaking, one of the most difficult things you do every day is to actually get up in the morning. And this is because you've just spent the entire night laying supine, and then all of a sudden you stand up at the end of the night. And when you do that, all the blood is going to rush out of your head unless you've got this barrel reflex that basically keeps the blood in your head and increases your blood pressure and keeps the blood up there. And that requires a lot of energy. And so cortisol is providing that energy. It's activating glucose. So you've got a lot of glucose, so you've got energy to do this. So if you get up in the middle of the night and stand up quickly, one of the things that you can experience is some lightheadedness. And that's because you don't have enough glucose to activate this response quickly enough. And so again, this is one of the things that cortisol is doing, is that it's anticipating when you are supposed to be waking up. It also means that if you're not sleeping at the right time, this isn't happening at the right time. The circadian clock, the way that you experience this probably the most acutely is when you travel across time zones. When you travel across time zones, that jet lag feeling is basically the circadian clock not being synchronized to your current behavioral schedule. And the jet lag is this kind of feeling, and it usually takes several days for it to adapt. We usually estimate around 1 day per time zone traveled. But again, this will be mistimed with many other things in the brain and the body when you're sleeping at the wrong circadian time. And this is just a last one. There are lots of hormones, but this is just a third one to show you. This is growth hormone. Growth hormone, there are very low concentrations during the daytime. Women actually have some spikes during the day, but in men, it's typically very low. But it is very stimulated by slow wave sleep. And so during slow wave sleep, you've got very high peaks in growth hormone. And it's one of the thoughts of why infants spend so much time in slow wave sleep is because they need lots of growth hormone released, which is important for, well, as it says, growth. So those are some of the kind of the functional aspects of sleep and kind of what it's changing in the brain and the body. And now I'm going to kind of discuss quite briefly some of the neuroanatomy. So what is going on in the brain? How does the brain generate these states of sleep and wake? And frankly, it's not a single place. It's a very distributed system. These are some of the nuclei and neurotransmitters that are involved in controlling wakefulness. So you've got elevated release of serotonin by the raphe nuclei, of noradrenaline by the locus coeruleus, by dopamine, a little in the substantia nigra, but mainly in something called the ventral tegmental area, acetylcholine in the pedunculopontine lateral dorsal tegmentum, histamine in the tuberomammillary nucleus, something called hypocretin in the parafornical nucleus, and acetylcholine as well from the basal forebrain. So all of these things are elevated during wakefulness, and each contributes in a fairly unique way to what we experience as the wake state. except for hypocretin, if you lose any of these, if there's damage to any one of these, there's sometimes not a very robust change in kind of what you see. But let me go over some of kind of how these contribute, especially kind of from a more pathological point of view. So first of all, serotonin. So serotonin, again, is involved in sleep-wake. It's high during wake. It's very low during non-REM, and it's very, very low during REM sleep. Most antidepressants have whether people want them to or not, have an effect on serotonin, and that's going to affect sleep. So most antidepressants will have an effect on sleep, especially on REM sleep. And so one of the things that you see is in individuals who are on antidepressants often complain about having no dreams or strange dreams or oddly timed dreams. So it's often that they're reporting issues with dreaming. But most all, well, any of the serotonin-based antidepressants, but even most of the antidepressants in general will have some sort of effect on sleep. Noradrenaline, again, high during wake, low during sleep. One of the key things about noradrenaline here, I want to point out, is that they're in ADHD, one of the hypotheses is that you've got your noradrenaline is too low. There are many teens that are diagnosed with ADHD, but any of you who have, been around teens, have teens, remember your teenhood, remember that most teens are actually pretty sleep deprived. And when you take a look at the diagnostic criteria for ADHD, of inattention, hyperactivity, and impulsivity, these are basically the behaviors observed in every single sleep deprived teen. And so there's a genuine question out there about how many of these diagnoses of ADHD are genuine diagnoses of ADHD, and how much of them are actually complete misdiagnoses, and we're basically medicating teens who are sleep deprived, and that the actual treatment would be to get them to get more sleep as opposed to give them stimulants during the daytime. So this is still an outstanding question in the field, but one that I think is incredibly important. Dopamine, again, dopamine's high during wake, low during sleep. If you take a look at individuals who have Parkinson's disease, which has a dopaminergic dysfunction at its root, they have, in many of them, there are profound disruptions in sleep and wake. And it's, again, it's very difficult on caregivers, and you see a very similar thing in individuals with acetylcholine, and I'll show you an example of that in a moment. Histamine. So histamine is a neuromodulator. It's present in the body. It's present in the brain. It's in the gut. In the body, it's used in mast cells for immune function. In the brain, the tuberomammillary nucleus is the sole neural source of histamine. And exactly its role, we're not sure, but we do know it's super important, because if you take a look, antihistamine drugs are one of the most commonly prescribed drugs in hospitals, because their activity is to kind of inhibit the histamine receptors, and this will induce kind of a nice, easy state of sleepiness, and so kind of cuts the edge in a lot of anxiety for hospitalized individuals and helps them sleep. Unfortunately, most histamines or most antihistamines are called antihistamines, but they actually aren't just antihistaminergic. They also have lots of other functions, and they have significant cross-reactivity with the acetylcholine system and have lots of anticholinergic effects, including urinary incontinence is one of the more uncomfortable ones. And in addition to the anticholinergic effects, the antihistamines also work as they are often prescribed on the immune system. And they, antihistamines are commonly prescribed for people dealing with allergies. So do you really want to be giving something to help someone sleep when it's having such a large impact on the immune system? So we'll kind of get into that in a moment. Okay, so there's a question in the chat. A lot of them are used as sleep aids from Sumana. Are you talking about the serotonergic drugs?
Yeah, I didn't know if like the things like pseudoephedrine that you get in NyQuil, I wasn't sure where it falls, but it's also an antihistamine,
Yes.
or at least you take it for allergies.
Yes, yeah, and that's, so yeah, you can take it for allergies and you can take it for sleep, and they do work for sleep. It's just not a great solution for it, especially long term. And I'll get into the pharmacology a little later as well. So here we're going to take a look at narcolepsy. Narcolepsy, again, is one of the few things where if you get rid of hypocretin, and well, if you get rid of hypocretin, you get a fairly robust and dramatic phenotype. So this is a dog that doesn't have hypocretin, right? This dog has a canine version of narcolepsy. If humans don't have hypocretin, then they have narcolepsy. And so I just like the dog. The dog's blue. It's a very pretty dog. And what's happening here, if you don't have hypocretin and you have narcolepsy, then you can have something called cataplexy. And this dog is experiencing cataplexy. And cataplexy is a gradual loss of muscle tone with a gradual increased suppression of spinal motor neurons. So basically what's happening is that thing that's happening normally during REM sleep, the suppression of motor activity, is now happening during wakefulness. And it can be triggered by pleasurable events. So typically in humans, it's triggered by laughing. In dogs, it's this is called the food elicited cataplexy test. In dogs, this is triggered by yummy food. And here's the dog. The dog's totally paralyzed. The dog's name is Blue, but she's awake. She's completely awake and she can't move at all. So this is what happens when you have a cataplectic attack. You're awake, but you can't move. You're completely paralyzed. The other thing that happens when you lose hypocretin is your sleep and wake become highly fractionated. And so it's very difficult to stay awake. You can't stay awake for 16 hours and then sleep for 8 hours. You can sleep for a couple hours, you can stay awake for several hours, but you cannot maintain this kind of 8 hours of sleep, 16 hours of wake. And so these are kind of the only one where when you lose it, that you have this truly, really traumatic impact on sleep-wake behavior. When you lose acetylcholine, which is what happens in Alzheimer's disease, you get a very similar thing that happen in Parkinson's disease. This is a plot that we can typically plot. This is 2 days in a row, and then days are stacked on top of each other. The black lines here are movement. The yellow is bright light. But this is called an actogram. This is kind of similar to what you might see with a Fitbit or an Apple Watch. This is the kind of data that it records. It's basically looking at wrist movement. If you see long periods of undisturbed movement, like here and here, this is sleep. So this is an individual over the course of a week with Alzheimer's disease. And what you can see here is that they're basically kind of randomly awake and asleep across the day and night. And this is not uncommon in individuals with Alzheimer's. In fact, on survey studies, it is the #1 or #2 reason for institutionalization of individuals with Alzheimer's disease. And what happens is that their caregivers, being either their children or their spouses, just can't deal with the constant disruption. So if the caregivers are children, it's often that the children are worried that their parent is going to be wandering because they're up in the middle of the night. They have dementia, so cognitively they're not altogether there. And this is very concerning to them. If it's a spousal caregiver, that's a concern. And there's also a concern because this fatigue during the daytime also leads to social isolation because they are unwilling or unable to go out as much during the day and be social, which leads to further isolation. And that can lead to further fragmentation of sleep and wake. And so again, these often happen prior to kind of the Hollywood version of, people can't remember if they ate lunch or who you are. So long before that stuff happens, individuals are often institutionalized because of these sleep-wake disruptions. The other reason that depending on the population you're surveying, that is is for incontinence. And that's the other reason for institutionalization. These two reasons, anyway, in different surveys, are reasons for institutionalization long before kind of the memory loss issues. So this is thought to be, again, due to the loss of cholinergic activity in Alzheimer's, and you get a similar, though not as dramatic version in Parkinson's disease. In Parkinson's disease, as well as Alzheimer's, you often also get some issues with emotion regulation during this time, which makes them even more difficult to handle. So again, all very problematic. There are lots of other ways that wake is regulated by the brain. This is just kind of a little laundry list. I have to say, 20 years ago, we didn't know that hypocretin existed, and now we realize it's a major player in how wake is regulated. Here are some other things that likely impact wakefulness, and there's probably other things on this that we don't have on the list yet, that we just don't know that it actually has an important role in terms of regulating wake. So non-REM is a little more straightforward. Basically, what's happening in non-REM is you have a nucleus in the brain, in the hypothalamus, called the VILPO, or the ventrolateral preoptic nucleus. And the ventrolateral preoptic nucleus is a GABAergic nucleus. So it contains GABA, which is an inhibitory neurotransmitter. And this projects to most of the parts of the brain that are involved in stimulating wake. So what happens is that this suppresses all of those wake promoting areas. And so GABA you see turn on at the beginning of the night when people are falling asleep, and it suppresses all of these wake promoting areas. And then that allows for a shift into a sleep state. So that is one of the key areas. Another one is adenosine in the basal forebrain. Let me see. Yep. And adenosine, we think it's, I should say, we think it's in the basal forebrain. We're not 100% sure. Adenosine is released as a consequence of cellular metabolism. So, when the cell metabolizes things, it uses energy. That energy comes from ATP, which is adenosine triphosphate. ATP goes to ADP, which then goes to AMP, which then goes to adenosine. The adenosine is then released from the neuron, it gets picked up by the glia, and then gets reconstituted into ATP and shuttled back to the neurons. So as the day goes on, you have a greater and greater increased rate of adenosine release. And this adenosine release then outstrips the ability of the cells to pick it up. And adenosine is an inhibitory transmitter and will cause cell firing to slow down. And so basically the thought is that as adenosine kind of, you get more and more adenosine in the synaptic areas across the day, and this starts to inhibit the cholinergic activity in the basal forebrain. Now, as I said, it's probably in the basal forebrain. We're not 100% sure, but we definitely know that adenosine is a key player in regulating sleep and wake, because at physiologic concentrations, the main impact of caffeine is to block adenosine receptors. And so for everyone drinking that cup of coffee, this is how it's doing it. It's blocking these adenosine receptors. It's blocking this impact of adenosine on its receptors and its inhibitory ability, its inhibition of the cholinergic activity. Now, caffeine is something which is kind of complicated. And one of the main reasons that it gets complicated is because the half-life in healthy people varies tremendously. It can have a half-life variation between 2 and 12 hours, which is enormous, which means that one person has a cup of coffee with breakfast, and by the time lunch comes around, they've got no caffeine in their brain. Another person has that same cup of coffee with breakfast, and by the time they're trying to go to sleep, they still have around half a cup of coffee running around their brain. Common things like oral steroid contraceptives, they double half-life of caffeine. So if you had an 8-hour half-life in your caffeine, now it's 16 hours. And then nicotine cuts it in half. So if you had again, in 8 hours, now it's 4 hours. So there's lots and lots of variation. And this is one of the reasons why, some people seem to be able to have, 3-4 cups of coffee in the morning and really have no problem at all falling asleep. And other people have 1 cup of coffee in the morning and they say it still bothers them at night. Well, it really can still bother them at night. And a lot of this can be mitigated by, I should say, sleep deprivation. You know, we studied caffeine in Stanford undergraduates, and they were downing a couple of Red Bulls and going to sleep just like that. Not a problem. They are so sleep deprived, it just doesn't matter how much caffeine they had. So anyway, that's kind of the role of caffeine in there. So Ulissa asks, in individuals with Alzheimer's, do their disturbed sleep cycles have anything to do with sundowning? Definitely. I have to say sundowning is a very controversial area. The disturbed sleep cycles probably do, though we are not sure exactly how they are involved. So in sundowning, you get a lot of agitation in the evening in individuals. That agitation may be secondary to them trying to stay awake in the evening. It's kind of maybe a phenomenon related to paradoxical hyperactivity, which is when some individuals get tired, especially you see this in kids. When kids get tired, they often become hyperactive. And this may be a kind of similar phenomenon, which is basically you're tired, you want to go to sleep, but then part of the brain saying, no, you got to stay awake. And so it agitates you into a state of wakefulness. This may also be happening in individuals who stay up too late at night. They basically can stay up kind of past their window. So what ends up happening is that you have a situation where if you have a fairly regular sleep timing, the hours before you normally fall asleep, it is very difficult to fall asleep because you have this big increase in circadian alertness during these hours. So if you normally go to sleep at 11 o'clock at night, if you're trying to go to sleep at 8 o'clock at night for some reason, or if you're traveling and your body's still set at the wrong time, you're trying to go to sleep, it's very difficult to fall asleep early because the circadian system is telling you to be awake. And then if you stay up too late, what happens is the brain says, oh, you're awake. It's past your bedtime. There's got to be a reason. So let me start increasing release of various kinds of neurotransmitters to try to keep you awake. And then you become agitated in a state of wakefulness, which is not a normal state of wakefulness. And so again, it creates this window of opportunity when you can kind of naturally fall asleep without either the circadian system telling you to be awake or these agitation signals kind of telling you to be awake. Okay, so again, as with wake, there's lots of other things involved in the regulation of non-REM. This is just kind of a laundry list. There's probably other ones, again, that have not been discovered. And the relative roles of each of these things, again, is not particularly well understood. REM sleep is very similar to what's happening during non-REM sleep. The main difference between REM sleep and non-REM sleep is acetylcholine, which is that there's a reduction in suppression of acetylcholine in the basal forebrain and the pedunculopontine lateral tegmentum. And this reduction in suppression causes the acetylcholine levels to go up a little. And that shift in acetylcholine levels is then able to shift the brain into this REM-like state of firing. And so that's the main difference between REM and non-REM in terms of what's happening at the neurobiological level. So I do want to talk briefly about melatonin. There's always questions about melatonin. Melatonin is produced in this near square wave fashion that you can see here. This is over the course of 32 hours. These are the concentrations. These are in young men. You have a similar pattern in young women as well. But the dotted line here is the time of typical bedtime. The solid line here is typical wake time. You get a similar pattern if you're awake or you're asleep. This is under strict circadian control. So the circadian clock is telling when melatonin is supposed to go up and when it's supposed to go down. Excuse me. Melatonin is suppressed by light, but otherwise will have this kind of pattern. The melatonin may contribute to sleep onset. It might not. We've studied people who don't make any melatonin, and it doesn't really seem to make that much of a difference in their sleep. If it were involved, we're not exactly sure how it's involved. It could be direct. There are melatonin receptors all over the brain, including in the circadian clock, which is in something called the SCN, or the suprachiasmatic nucleus. And so it might be that this melatonin at the beginning of the night rises and suppresses this circadian alertness signal. It also can act peripherally on vasodilatory mechanisms to increase the amount of heat loss that you get. It can also do that centrally in the hypothalamus, so it might act on heat loss, but basically we're not exactly sure. And a little later, I'll talk about taking melatonin, so kind of the impact of exogenous melatonin. But in terms of endogenous melatonin, it might have a role, but it's not going to be that big, at least in humans. In non-mammalian vertebrates, it's a much more potent sleep signal. So far I've been talking about sleep and what sleep is, but now I'm going to talk about kind of how sleep can go wrong. So the first kind of major sleep disorder that I want to talk about is sleep apnea. So sleep apnea is a condition in which you have repeated blockage of the upper airway, which leads to a brief hypoxia, an awakening, a gasping breath, and a return to sleep. And so basically you see here kind of at the back of the airway, you get a compression and you're unable to breathe, and this leads to these hypoxic events. In someone where it's mild, you're talking about having only around 40 awakenings a night. Moderate, 120. Severe, you're talking about 400 awakenings per night. That's basically every other minute you are being choked into consciousness. It happens in about a quarter of adults, and most are unaware. Obesity is a significant risk factor, as is older age. Being male is a big risk factor. Being female is protective until you're postmenopausal. Once women are postmenopausal, then they have the same risk factor as similarly aged and BMI males. And the use of muscle relaxants, such as alcohol and some drugs, can also increase the risk of sleep apnea. Now, one of the things is that, all right, you're waking up 400 times a night. It seems like you should be aware of that. And one of the problems is that you don't have memory of waking up when it's brief. And this is just a little experiment that was done many years ago, basically showing that you don't make the memories of when you fall asleep. And anyone who has bad sleep, I beg you, please don't do this. It will make your sleep worse. But if you have really good sleep, go ahead, feel free to do this. Try to remember that moment when you're falling asleep. And you can't do it because the brain doesn't make that memory. That memory disappears. And it can be anyone from 1 to several minutes. You can wake up, have a phone call, have an entire chat, fall back asleep, and have no memory of that phone call. When I talk to medical residents, I always tell them, if you're on call, write down whatever you think you're saying, and you'll be surprised in the morning how many times you don't even remember having that conversation. It's your handwriting and you wrote the note. And so anyway, so that's one of the problems, that you can wake up here, 200-300 times a night. Each time you wake up, take a gasping breath, you fall back asleep, no memory of that. And so oftentimes people who have sleep apnea, their sleep is very fractionated, and so they're very tired during the day. And so often they think that they have great sleep because when their head hits the pillow, they fall asleep like that because they're exhausted. They sleep 8 hours, or at least they think they sleep 8 hours, and they're exhausted all day. And so oftentimes they'll go in, they'll see an endocrinologist. What's wrong? I don't have energy. Oh, you should do this, you do that. They'll go see a neurologist. What's wrong with me? And finally, they'll end up seeing a sleep doctor who takes around half a second to diagnose them with sleep apnea. Colleagues of mine who work in the sleep clinic will say, a lot of times you tell the individual, you hook them up in the sleep lab. You can tell them, okay, let me just get out into the control room, and then we're going to run through some calibrations. And by the time you leave the room, they're already asleep. falling asleep in under 10 minutes would be considered pathologically sleepy. So that's one of the reasons that it's brought to kind of diagnostic attention. The other reason is what is called in the field as sleep divorce, which is that these closures in the back of the throat often result in snoring. So not all snoring is apnea, and not all apnea has snoring. But if you hear someone snoring, it's usually a pretty good indication that they've got apnea. Again, it's not perfect, but it's pretty good. And if they're not snoring, again, it doesn't mean they don't have apnea. But you have situations where basically it's you're going to go get this fixed or we're going to get a divorce. And you're sleeping on the couch because I can't take all of the snoring. So these are two of the reasons why it is often brought to attention. Standard treatment is weight loss because again, it's high BMIs are often associated with having sleep apnea. Positive airway pressure is the most common kind of treatment that's interventional. This is basically where you're putting a mask on someone's face during sleep, and it is providing what is called a pneumatic splint. It's basically creating a kind of a reverse vacuum. It's pushing air in, causing the airway to stay open, allowing you to breathe. And then less common and really only suggested under specific circumstances are various kinds of surgical changes, including changing the jaw structure or changing the content of the throat. They basically take a roto-rooter to the throat and get rid of a lot of the tissue. Many of these, it's very hit and miss on whether the surgical ones can work. There's all sorts of new treatment algorithms. People hate CPAP, which is a constant positive airway pressure. They really don't like it. It's effective when it's used, but it's often not used because people really don't like it. And so people are trying all sorts of different things, trying to look at different ways of treating sleep apnea. But CPAP is still kind of the gold standard in terms of treatment of sleep apnea. And I should mention, so yes, let me go on. So another very common sleep disorder is insomnia. Depending on exactly how you're defining it, anywhere between 10 and 30% of the population has insomnia. It's highly comorbid with other neuropsychiatric disorders, including anxiety, depression, PTSD. Risk factors include being female, older age, and occupational status, especially having a non-traditional schedule. These are all things associated with having insomnia. When we define insomnia, there is an official definition, which is the difficulty initiating, maintaining, or reinitiating sleep, which basically means you find it difficult to fall asleep, stay asleep, or go back to sleep, or you wake up too early. It has to happen 3 times a week for at least 3 months for it to be considered to be a chronic insomnia. It has to be where you have the adequate opportunity to sleep. It's not due to some other sleep disorder or substance. And importantly, it has to have some sort of daytime distress or impairment. And notice there's no objective criteria on sleep time, which means that if you get up for an hour in the middle of the night and it doesn't bother you, that's not insomnia. If you wake up 4 times a night and you can fall back to sleep and it doesn't bother you, that's not insomnia. If you wake up for 10 minutes in the middle of the night and it causes you anxiety, or it bothers you, or causes worry, that's insomnia. So again, insomnia, how it's defined, is not just about your sleep, but it's more about how you think about your sleep. It's how you treat your sleep, how you're worried about your sleep. Like, oh, I'm not sleeping, I'm going to be wiped tomorrow, I'm not going to be able to do my job. These are some of the issues that come up in insomnia. Again, it's not how much you're sleeping, it's your perception of that sleep. Okay, so heading back a second. So Lori has a question: is sleep apnea a risk factor for Parkinson's, even if the person subjectively has no problem sleeping? I don't know if it's specifically for Parkinson's. Sleep apnea, I should say, has, and I should have mentioned this, has some fairly significant it causes some fairly significant damage to the brain and to the cardiovascular system. So these repeated hypoxic events, these repeated awakenings, which are sympathetic events, seem to cause, or seem to be associated at least, with cardiovascular damage, increased risk of cardiovascular disease, heart attacks. There's memory loss. There seems to be other kinds of cognitive impairments. I don't know the answer if it's specifically associated with Parkinson's. I would not be shocked. Again, it's a fairly substantive damage that's being caused by this. And I'll get into that in a little as well. Okay. So insomnia, big societal impact, estimated $60 billion in lost productivity due to insomnia. You've got insomnia leading to sleepiness, which causes accidents at work, accidents on the road, errors, something that in the field we call presenteeism, which is someone shows up to work, but they're really so tired that they can't focus and they're not doing a good job, and a reduced quality of life. Basically, anyone who has insomnia will tell you, however you define it, it sucks. It really is very disruptive to everything about your life. As I mentioned before, it's got a very strong relationship with psychiatric disorders. This is a study that looked at about 1,000 people, and these people didn't have depression, anxiety, alcohol abuse, or alcohol use disorder or drug use disorder at baseline, and then they looked 3.5 years later. If they didn't have insomnia, that's in the red. If they had insomnia, and this was at baseline, it's in the blue, and you can see huge differences. So, only 5% of people had depression in the next 3.5 years, but if they had insomnia, that was 16%. Anxiety was doubled, alcohol use disorder doubled, and drug use also increased. Well, it's a very low amount, so it's hard to say, but increased tenfold. So having insomnia is really is not good for the development of other psychiatric disorders. There are many different ways to kind of think about insomnia. This is just one of many different models. I didn't want to kind of go into all the different models, but there are many different models. This is one of them. It's one of the older models. It's called the 3P model. The idea is that there are predisposing factors, precipitating factors, and perpetuating factors. Predisposing factors are basically that's who you are. It's your genetics. It's how your brain is wired. That's just, do you have a brain that's going to be more or less likely to have insomnia? Then there are precipitating factors. These are events that occur that can kind of trigger having insomnia, especially for the first time. And then there are perpetuating factors. And perpetuating factors are behaviors that you do that can basically make insomnia worse. And so this is kind of a kind of little model where you've got this, again, you've got your predisposing factor, something happens, and then you get insomnia. And then what happens, the impact of what happens kind of diminishes, but then is replaced by things that you're doing to compensate for this. So, for example, your pet dies, you're really upset, you can't sleep, and then you start, watching Netflix in order to fall asleep. And this is now giving you this bad habit of now you're associating that the only way you can fall asleep is to watch Netflix. And now all of a sudden that's not working anymore. And now you start developing other behaviors to compensate for that. And that, so then that gets into in this chronic phase. So anyway, this is just one of the models and just kind of one of the ways to think about the three different factors that are kind of involved in this. Smita asked about the previous slide. Could it be possible that alcohol and drug use are a contributing factor in causing insomnia? In those individuals, they didn't have that premorbidly. So they started out with no alcohol use disorder or no drug use disorder. beforehand. So they didn't have any of those predisposing factors at that point. Okay, so we talked about insomnia, but I have to say that, you know, for a long time, the field viewed sleep as kind of this binary structure, which is you had insomnia or you had good sleep. But most people realize that there's something in between, that insomnia is probably just one end of a continuum. And there are a lot of people who might not have good sleep, but they don't have insomnia. And so it's somewhere in between. And so Dan Bicey, a colleague at the University of Pittsburgh, has come up with something called the SATED model of sleep health. He's very good at these acronyms. Very good scientist, but also good at acronyms. And in the SATED model, there's kind of 5 ways that they're defining like how good your sleep is. One is like how satisfied are you with sleep? So basically subjectively, like how did you sleep last night? How good is it? How sleepy are you during the day? So basically functionally, how good is it? How consistent are you in your timing? How often do you awaken at night, and how long do you sleep? So these are kind of 5 factors that are involved. And you know, if you take a look at it, while you know insomnia itself is associated with lots of bad things, you know, having problems in any of these 5 areas are also, at least at the epidemiologic level, associated with bad outcomes as well. So if you take a look at if you have problems in any of these, again, people, you know, live less long. They've got coronary heart disease, depression, diabetes, obesity. So you've got lots of different associations of these things, basically saying if you're not having good sleep, you're also having other problems. So again, even if you don't have perfect sleep, it doesn't mean you have, so if you don't have perfect sleep, it doesn't mean you have insomnia, but it doesn't mean that you necessarily have good sleep. And there are lots of things that you can do to kind of help improve your sleep. And that's kind of what we're going to talk about now, is kind of how do we treat these sleep problems. And especially pointedly, how do we going to treat kind of insomnia is kind of where we'll start. And so there are 2 main approaches. One is a pharmacotherapy approach, and the other is something called cognitive behavioral therapy for insomnia, or CBTI. So in terms of pharmacotherapy, you know, benzodiazepines are still a great choice for short-term treatment. You know, your pet dies, you just don't want to be awake, you can take a benzo. Benzos really work well. They put you into a state of not being awake. Now, they do not recapitulate normal sleep, but they do what they are advertised to do, which is make sure that you are not awake. There are changes in sleep architecture. Again, you have lower slow wave sleep, there are carryover effects, there's abuse and dependence issues, discontinuation effects, lots of problems. But under, you know, a doctor's, care, they can be a very effective treatment. Several years ago, something called BZRAs, or benzodiazepine receptor agonists, came out. Ambien, Sonata, Lunesta. They're advertised all over the place. You've probably heard of them. They're called the Z drugs, zolpidem, zaloplan, and zapaclone. They're basically the same thing as benzos. They're not technically benzos, because you can see here, they don't have a benzene ring fused with a diazepine. And that's how you define a benzodiazepine. So they're technically, from a chemical standpoint, not a benzodiazepine. So benzodiazepines got really bad rap back in the 80s and into the 90s. There was a lot of abuse. There was a lot, you know, they were not prescribed well. So these drugs came out and they're like, look, these aren't benzos. They work great. And people got very excited. But basically, they're benzos. They don't look like a benzo, but they act on the benzodiazepine receptor, doing the exact same thing. So I'm not saying you should take one or the other. I'm just saying don't be fooled to think that they're not a benzodiazepine. They are. And they have very, very similar pharmacokinetic profiles. So another way, antihistamine. We talked about this briefly before. They're very commonly prescribed. But again, problem is they're anticholinergic. Most of the over-counter sleeping aids have some form of antihistamine. Diphenhydramine is one of the most common ones. And again, they work, but they have anticholinergic and immune function as well. So probably not very good for long-term use. There are 3 different kinds of histamine receptors. There's the H2 receptor, which is in the gut, H1 receptors, which are all over the brain, as well as on immune cells. And then there are the H3 receptors. Now, H3 receptors are on these TMN neurons, which is the tuberomammillary nucleus neurons. This is the only place in the brain, or the only place in the brain, the only neural source in the brain that histamine comes from. So H3 receptors are something called auto-inhibitory receptors. So basically, as the TMN releases histamine, the histamine binds to these H3 receptors and shuts down the TMN, reducing histamine. So they act as kind of a brake on this. So if you're producing too much, it slows it down. So the idea is that if you can activate this, you can reduce histamine and thereby increase the likelihood of falling asleep, because you're reducing this wake promoting system. The problem is that developing something which is an agonist is very difficult. Making something which is an antagonist or blocking the receptor is much easier. And that actually just came out, something called pitolisant or Wakix. This actually blocks this receptor, which increases histamine and is a wake promoting drug, and is being marketed as a wake promoting drug. But hopefully, I know people are working on developing H3 agonists, because again, that could decrease histamine production, and thereby, by decreasing a wake promoting something, basically allowing natural sleep to occur. I mentioned briefly SSRIs. SSRIs like fluoxetine block the reuptake of serotonin. This blockade, so sometimes sleep problems, especially long-term ones, are treated with low dose of antidepressants, changes the serotonin profile. Again, you often have REM suppression. SNRIs have the same problem. So these are specific norepinephrine reuptake, serotonin and norepinephrine reuptake inhibitors. They block both the reuptake of norepinephrine and serotonin. Again, they often have REM suppression in low doses, can often be used to treat sleep problems. And then you have SARIs, which both block the reuptake of serotonin and block the 5-HT2 serotonin receptor. These tend to have less of an impact on REM sleep and enact as hypnotic. So again, these are some treatments, and again, you often have an association of depression with insomnia. So sometimes you're double dipping and having an effect on both. And now you've got this other system, hypocretin. Now, if you remember, hypocretin, if you lose hypocretin, you get narcolepsy. But what hypocretin is normally doing is that hypocretin is normally acting as a circadian wake drive. So basically, the circadian wake drive, this is just kind of a picture of it. This is your normal bedtime, normal wake time. It's peaking. This is taking, how long does it take you to fall asleep? It's peaking right before your normal bedtime. This is being caused by hypocretin. So the question is well, if you block it, what will it do? So if you kind of block this, what will it do? And so there are now 3 different hypocretin antagonists that are, well, 2 that are on the market, one that's coming, that people use for insomnia to kind of block this wake drive. They're not that effective. People use them for insomnia. They're not great, but where they are effective is during the daytime. And so basically, if you are blocking the circadian wake signal, you're blocking this hypocretin during the daytime. Now, you're not blocking it so much that you're giving someone, you're making someone a narcoleptic, but you are blocking it sufficient to reduce the amount of hypocretin that's there. What you get is a very effective pill for daytime sleep. So this is a study that we did in JAMA a couple of years ago, basically showing that what you can get is, if you're comparing suvorexant, which is one of these medications, with placebo, that you have these are in shift workers doing random shifts and random sleeping times, but the only instruction is you take one of these pills before you sleep during the day, and their sleeping time went from 4 hours to 7 hours. So they had a 2 to 3 hour increase in total sleep time. And so if you are blocking this signal during the daytime, this is what is preventing shift workers from sleeping. So if you are trying to sleep during the day and you have an adequate opportunity, what happens is that you can sleep like 4 hours because you're tired. And then once you burn off that need, that homeostatic drive for sleep, you burn most of that off in the first 4 hours. Then not only do you not have that drive, but you now also have this drive for wake coming from the circadian system. And this is why, you can sleep, you go to sleep at like 10 o'clock, and then at 2 o'clock in the afternoon, you're up and you can't sleep anymore, even though you're still tired. So this can kind of stretch it from 4 to 7 hours. And it's also something that we think, that we haven't tested it yet, that should be very effective for work, to work in individuals who are jet lagged. Another thing that people use, and I said I'd circle back to this, is melatonin. So there's all sorts of agonists. People can use actual melatonin. It comes in you can go to the grocery store or CVS, it comes anywhere from 0.3 to over more than 10 milligrams. There's also bromeltein and tazobmeltein, which are agonists. Agomelatine, which is an agonist, is a dual agonist and also a 5-HT2C receptor agonist. And you can use all of these. The problem is that it's not a particularly effective hypnotic at night. Some people love it, and if it works for you, great. But when you give it in a placebo controlled double-blind study, you see that it might shorten sleep latency by a couple of minutes. Doesn't have any effect on total sleep time, and again, just doesn't work very well at night. During the daytime, it can give you an extra half hour of sleep. If you're traveling, it can give you some extra sleep as well. But if you're taking it at night as a treatment for insomnia, it's not a great hypnotic. The other thing to consider, one is that if you're taking melatonin, make sure that it is tested at all for purity and potency. This is one of many studies that basically pulled melatonin off the shelves at drugstores and tested it with HPLC, and basically see there's lots of contaminants in these commercial formulations. Melatonin is the only hormone that is not FDA regulated. Because it is present in nanogram quantities in rice and bananas, it is considered a nutraceutical. And so it is not FDA regulated. So there's no FDA regulation on what is in these pills. So just be careful in what you're taking. The other thing to consider here is that this is, around 100, just under 100 picograms per mil is normal nighttime levels. It's even high in a lot of people. If you take 0.3 milligrams, that will give you high levels at night. If you were to take 1 milligram, it gives you pharmacologic levels. If you take 10 milligrams, it gives you super pharmacologic levels, huge levels of melatonin if you're taking 10 milligrams. So high that when you take it, if you're taking it for sleep, that you have super high levels of melatonin when you wake up in the morning. In fact, if you're taking 10 milligrams of melatonin at night, every night, you have very high levels of melatonin all the time. If you're taking melatonin to help shift your circadian clock, it's not as effective as light, but people use it. You take the low dose, because if you take the high dose, it ends up being ineffective. But if you take a dose under 0.3 milligrams, that can be effective. And the lower the dose, actually, the better, if that's what you're using. So anyway, that's just stuff to consider about melatonin. Getting into cognitive behavioral therapy for insomnia, CBTI it's really it's a toolbox. As I mentioned before, how people's sleep is bad, how people's insomnia affects them, it's very personal, and it's different in everybody. And so what CBTI does is it provides different ways of attacking this. And so you have a behavioral component, you've got a cognitive component, and then you also have an education component. It's very effective. It's as effective, if not more so, than pharmacotherapy, especially long-term. It's compared to other psychotherapy, it's brief, it's 4 to 8 sessions. This is not, this is not psychotherapy. This is not, lie on the couch and tell me about how your mother screwed up your sleep. This is very much more functional kinds of therapy. And so basically the first thing, is this kind of to understand is that CBTI usually does not change your sleep. If you take a look at the objective amounts of sleep that people are getting before and after, there's usually not much of a change. If you ask them about their sleep, they're like, yeah, it's great. I haven't slept this well in years. Do you have insomnia? No, I don't have insomnia. You do questionnaires, don't have insomnia, but they're not sleeping anymore, but they feel better about it. And the reason is that you're basically changing an individual's perception about their own biochemistry. And so I'm just showing this picture here of people training to do free diving. If you're training to do free diving, you're basically learning how to ignore your body's signal of needing oxygen, right? Your CO2 levels are going up, and you are learning how to ignore these signals. It's still happening, but you're ignoring them. And this is kind of what you're doing with insomnia. You still have all of these biochemical signals going on in the brain, and you're just learning how to ignore them and kind of living with them and just saying, okay, they're still happening, but you just don't care as much anymore. And again, that's easier said than done. And so to do this, again, these are kind of the parts, and these are behavioral targets of stimulus control. And the idea is that what you want to do is you want to say, okay, you want to set up a pattern that when you get into bed, you fall asleep. And that's a good thing. It's not that you get into bed and then you struggle to fall asleep, or you get into bed and you get anxious, that you get into bed and you fall asleep. And so basically there are all sorts of things you're doing to make this association. Getting to bed only when sleepy, and ready to go to bed, only using the bed for sleep and sex, getting up at the same time every morning, getting out of bed if you can't fall asleep. Again, trying to associate this bed with good things. You can also use sleep restriction, which is basically where you start reducing people's sleep at night to get them tired, to kind of add a little push to make it a little easier to fall asleep. And you can't have any napping, because again, if you're napping, then you're not going to be as tired at night. So again, the idea here in sleep restriction is you want to increase this drive for sleep, so that when someone gets into bed ready to fall asleep, that there's a little extra something in there to help them fall asleep. Sleep hygiene in and of itself is insufficient, but for many people with insomnia, many parts of it help. So again, it's talking about caffeine, nicotine, exercise, diet, your sleep environment. Again, all of these things can have an impact on sleep, especially in people without insomnia. So a lot of times you'll have someone who comes in, and I'll be talking to someone who is in they're 85 years old, and you start talking to them about their sleep, and they're like, yeah. You find out, oh, they have a cup of coffee every night after dinner. You're like, well, caffeine can impact your sleep. And they're like, well, it's never impacted my sleep before. It's like, well, as you get older, these systems start to fail. And that cup of coffee that didn't used to impact your sleep now can. Again, when you smoke, if you smoke, that can have an effect. Exercise is very complicated, but exercise can have both positive and negative impacts on sleep in terms of the timing of the exercise relative to sleep. And the intensity of the exercise, when you eat and what you eat, can have an impact on sleep. How hot or cold it is in your room, if it's noisy, if there's light, all these things can have an impact, and there are various kinds of mitigation strategies that can be used for these that people are thinking about, like, well, if we can kind of monitor this, can we automatically, adjust it? Are we able to say, oh, you know what, that when the garbage truck comes by at 5 o'clock in the morning, it wakes you up. So what we're going to do is we're going to introduce white noise into the room before this happens, so that you don't hear that and it doesn't wake you up. There are also cognitive targets. So again, these are looking at targeting things like about why are you worried? How do you think about sleep in ways that might be true but aren't helpful? How do we reduce your attentional bias? So this is where you start fixating on things that are keeping you awake, and reducing things that again might help you. Reduce things that are going to help keep you awake. So again, going after the cognition, going after behavior. These are various ways that can be done to help individuals kind of get control over their sleep and stop worrying about their sleep. Again, easier said than done. The biggest problem with CBTI is this mismatch, is that you've got a lot of people with insomnia, and a lot of people, and even more people who don't have insomnia, just have bad sleep and could really benefit from CBTI. And the issue is that you just don't have enough people giving CBTI. I mean, just as an example, in the entire country of South Korea, there is one provider of CBTI. And I have to sadly say that one provider is no longer in Korea. They're coming and doing sabbatical in my laboratory. So currently, South Korea has 0 people who are qualified to give CBTI. If you're in the United States, you can get CBTI, especially if you live in a city. And now with Zoom, well, you can even do individual telehealth, and that's great, but there still aren't enough providers. Some people can do in-group, some people can do group, some people can do group online, some people can do apps. There's lots of apps that are out there. The problem is that not everyone is going to respond to an app. Not everyone is going to respond to group. Not everyone's going to respond to in-person. So the question is, can we develop some sort of triage? Can we figure out who's going to respond to what, and then give them the therapy that they need, and that's going to be effective? Because what you don't want to have happen is someone do an app, go, this app sucks, it's not working, and then never try CBTI again because they know it doesn't work because they did the app, even though if they did in person, it might really help them. And so I actually think that a lot of the apps are, while some of them work very well, but many of them work very well, that they are not prescribing to the do no harm, because they have not figured out yet who this might actually cause harm in. But hopefully that will improve in the future. There's lots of alternatives: self-hypnosis, biofeedback, acupuncture, herbal supplements, light therapy. And again, in different individuals, these work. As I mentioned with the melatonin before, even though in the double-blind placebo-controlled trials, melatonin doesn't work, if you take melatonin and you think it's working, that's great. You know, even if it's a placebo effect, doesn't matter. It's working for you. Keep taking it. So again, there's lots of ways that you can treat it. And now there are even some products that are on the market. There's one which does some forehead cooling, which if you compare, well, anyway, this forehead cooling, I don't know if it actually works. It is FDA approved, but the FDA basically did not approve it on effectiveness. It approved it on that it doesn't do any harm. There's, you can do bone conductance induced delta activity, which is basically you use sound waves that penetrate the brain and help increase this kind of those slow waves, those delta waves that you see during deep sleep. Whether it's effective or not, again, still remains to be seen. There's another product. This does light and sound induced flow state. I have to say that I advise this company, so that's my disclosure for this particular product. So I don't want to say any more about it, but they're doing clinical trials to see how effective it is. So there are some other things. You know, so how do we measure sleep now and in the future? Sadly, this looks funny, but it's true. This is what sleep is recorded in the lab. We put on lots and lots of electrodes and then tell you to go to sleep. This has been the status quo since the 1950s. Now, the devices have gotten smaller. They're no longer giant, you know, polygraph machines that take up, you know, a quarter of a room. Now they can fit in the palm of my hand. But still, we got lots of recording things. And we get one night because it's so complicated to do these kinds of recordings. So what else is out there? Well, you've got headband worn things. You've got devices that fit on the forehead. You've got ear worn devices. These all do single channel EEG and then automated scoring. So the scoring for this is typically by hand. You've got an individual who looks at the polysomnogram and says whether you're in wake and 1 and 2 and 3 are REM. That's being automated. This is all automated. You can get multiple nights, which is great. I'm not sure if it's actually useful. You know, if someone said they could record, you know, 50 nights of data and just showed me that data, I don't know what it would really tell me. So there's that. There's using movement as a proxy. So if you wear something on the wrist, like an Apple Watch or a Fitbit or a research grade device, you can do that. There are mattress-based ones that are in pucks or your phone or piezoelectric strips. These are all basically using movement as a proxy. Another thing that uses movement as a proxy is using low-powered radio waves, and this is actually called a nearable since it's not touching you. Newer additions are also including heart rate and heart rate variability into it. And the problem is that, you know, all these devices, they work okay if you're a good sleeper. So if you're a good sleeper, you know, it tells you your total sleep time pretty accurately. They don't tell you napping accurately. They don't tell you sleep latency accurately. They definitely do not report sleep stages accurately, even if you're a good sleeper. None of these things are accurate. If you're a bad sleeper, they don't even report your total sleep time accurately. So what it doesn't do well is it doesn't pick up wakefulness very well. So the more you're awake and the more fractionated your sleep is, the worse these things do. And you know, we've studied people with traumatic brain injury, and the polysomnogram was telling us they were sleeping like 3 hours, and the wearable was saying they were sleeping 7 hours. And that's because they just weren't moving around a lot. They woke up a lot, but they weren't moving around a lot. So these wearables thought they were asleep, and it wasn't the case. So again, if you're a bad sleeper, they're not very good. And the other problem is, you know, kind of, so what? You know, so if you look at it, you can think of it as kind of a 2x2 array, which is, you know, how do you feel and what is the device telling you about your sleep? You know, and you've got you felt like you had a good night's sleep and it's telling you it was good. You know, duh. Same thing with the bad. And if the device told you you had good, but you felt like it was bad, you're wondering, well, is this working? And then if you felt like it was good and the device is telling you it's bad, well, now it just gave you insomnia. So again, I'm not sure, as currently structured, if these, you know, wearables, you know, are useful. But I think that there is, you know, there's possibilities for the future. And, you know, I think that, you know, in the future, when we're looking at sleep, the wearables, you know, are nice, but frankly, a lot of them, people charge at night, so that's not very useful. A lot of them, people don't like wearing at night, so that's not very useful. So I think that really what's going to happen in the future in terms of collecting sleep data is that it's not going to require wearing a device. It's going to be some sort of nearable. It's going to be something that is going to passively collect sleep data for a long period of time. And then it's going to integrate this with collection of sleep outcomes. So how's your metabolism, your alertness, your cognition? It's going to integrate this with behavioral data. What are you doing? You know, are you exercising? What are you eating? What does your built environment look like? And it's going to be able to take these kinds of 3 different data streams and integrate them, and then actually give you useful feedback. It's going to be able to tell you things like, oh, you know, every time you go to that Mexican restaurant after 10 o'clock, you don't sleep well. Now, again, I don't know what you're doing. I don't know what you're eating, if you're drinking, if you're working. All I know is you don't sleep well. So that's actually actionable information. Or every time the room gets too warm, you don't sleep well. So can I tell your Nest thermostat to turn the temperature down at night? Again, that can be useful. You know, oh, look here, you know, we're monitoring, you know, it looks like, you know, you're getting 7 hours of sleep, but, you know, we're monitoring your daytime performance. And it looks like that when you get 8 hours of sleep, you actually increase your performance by 10%. I don't know if that's important for you, but that's what it's doing. Again, this is where it becomes helpful. You know, if you could detect, for example, sleep apnea before it gets to be a problem, that becomes helpful. So as I mentioned, sleep apnea causes all this damage to the brain, to the heart. But one of the causes that's causing all this damage is because it's not detected for years before the people reach the clinic. So the question is, well, if you were able to detect it earlier, could you then treat it earlier and prevent this damage? Because once the damage has occurred, it does seem to be not reversible. And that's a problem. So, you know, and if you're looking at these sleep problems, okay, well, what happens if we can intervene a little earlier? What happens if we can pick up that insomnia before it becomes chronic insomnia? What happens if we can, you know, start giving you advice or start having these changes before it becomes problematic, preventing all of this damage? And also thinking of sleep as a platform, saying, you know what, we're able to monitor sleep and things that are happening during sleep, and we might be able to detect things that we can't detect during the day because your behavior is basically hiding them. So for example, if you're having a problem in a particular hormone or metabolism, well, that's very difficult to detect during the daytime, given all of the different behaviors that you might have. But during sleep, we know what your behavior is. So again, I think that the future of sleep is a very positive one. I think there's a lot of things that can be done with these kinds of devices in ways that they can be integrated, but really we need this integration of information in order to make that better. And again, it's this idea of closing the loop, and this kind of, the more passive it can be, the more likely it is to work. So with that, I thank you, and I'm happy to chat and take some questions. It's just a picture of my lab, not a big lab, but we get a lot done, and of the new Center for Sleep and Circadian Sciences. So if you're interested in sleep and kind of what's going on here on campus, this is the new hub for all that kind of information, including our new seminar series, and then all the various people around campus that are doing sleep. So
thank you so much.
Thank you.
I really appreciate it. This was a very informative talk. We really appreciate it. I have a question here. Sumana is asking, can you talk about biphasic sleep? Is that a real thing?
Sure. Yeah. So there are lots of stable structures that your sleep and wake pattern can have. The ones that are stable are the kind of the 16-8 structure, so you're awake for 16 and then you sleep for 8. Another one is the siesta structure, which is you have a shorter night of sleep, but you get up early and then you've got a nap during the afternoon, an hour or two during the afternoon. And that is also a stable structure. And then there's also this biphasic pattern, which again has kind of had popularity several years ago. Roger Urkich, a historian, wrote a book about this. And in Northern Europe in the Middle Ages, this was a popular sleep structure and is a stable one. And this is basically Northern Europe, especially in the wintertime. It doesn't stay light out for very long. It gets dark early and stays dark late in the morning. And no electricity, and lighting is expensive. And so you had cases where people would retire near the time of sundown and get up around sunrise, but it's like 12 hours and you can't sleep that long. So they get up in the middle of the night, and you can get up for a couple of hours in the middle of the night, and that totally works just fine. So if you go to sleep early and you wake up in the middle of the night, spend a couple hours awake, and then go back to sleep, and then wake up around sunrise, again, that's another stable one. And it does appear probably limited in terms of its historical use in Northern Europe. If you people have taken a look at individuals who live in modern pre-industrial societies, and those individuals are typically not doing that. They are staying up. These are typical equatorial, so the nights aren't as long. They still go to sleep after sundown. They sit around the fire. They go to sleep several hours after sundown, and they wake up at sunrise. But what isn't stable is one of the popular ones that on the internet, at least, is the Uberman schedule or variants like that, where people are like, okay, I'm going to be up for an hour and sleep for 30 minutes. So you still have the same 1-to-2 ratio of sleep to wake, but they do it in like 1 hour segments or 3 hour segments. That's not stable. You can do it. It takes a lot of effort, but it is not a naturally stable thing. And it likely contributes to lots of sleep deprivation and starts to fragment sleep and wake. So I think that it may be an effective tool to increase artistic creativity, because I think one of the things that might be happening, it's not something that we can ethically test, but something that might be happening in this is that individuals are basically having REM sleep intrusion into their wake. So while they're awake, they're part of their brain's in REM and part of it's awake. And the way we view REM sleep and thinking during REM sleep is basically REM sleep is a state of controlled psychosis. So you are basically psychotic during REM sleep, but you're paralyzed and asleep, so that's okay. But if that's happening while you're awake, that may not be so good. So if you're looking for artistic creativity, maybe that's great. But if you're trying to solve a physics problem, maybe that's not so good. So again, that is not a stable form of sleeping weight. And then I see Susan has a question in the chat. Wearing an eye mask really helps my husband and I sleep longer on the weekends when we can sleep longer. Why the promoter? Yeah, so eye masks work great in some individuals. We call my mother-in-law the human photon detector. You know, there's like one stray light, you know, beam that comes into the room and she wakes up. And years ago, I set her up with an eye mask and she's been sleeping well ever since. So they're fantastic. If you are, you know, if you are someone who is, you know, awakened by light, and there are many people who are, you know, these can work great. For people who don't like wearing things in their face during sleep or bothered by an eye mask, you can also do blackout curtains. And there are lots of blackout curtains that are out there that can really help help your sleep. Because again, that kind of light in the morning, some people can be very rapidly awoken by that light.
Thank you. We had Lori raise her hand. Please feel free to unmute yourself and ask your question. Thank you.
Yeah, thank you so much, Dr. Zeitzer. Is that how you say it?
Yep.
Yeah, there was a lot that I learned from this, especially thinking about coming into menopause, especially with my memory consolidation. I'm going to use that with my staff because they're used to having to send me emails constantly to remind me of things if I don't write it down. So that gives me a little bit of self-compassion. Thank you for that. So what is the research then happening here at Stanford with perimenopause and menopause and sleep?
Sadly, so Lori, thank you for the question. So sadly, very little. There's some pregnancy research, but right now, no perimenopausal research. We had done, we did some, and we're still exploring it. But I have to say, middle-aged women and women in the perimenopausal phase are not studied very often for these problems. And one of the problems is that they actually have too much to do to come in and do these kinds of studies. So the vast majority of studies are done, you know, and you have to be careful with biomedical research. You know, so many of the studies that are on healthy basically means underemployed 18 to 30-year-old, typically males. You know, women too complicated because of the menstrual cycle. Oh, you know, can't study that. So it's very biased. The research is often quite biased. You know, we, for example, in this study, which we did, we found these big spikes in cortisol in women that were perimenopausal. About 10% of the women had these big spikes in cortisol in the beginning of the night that were associated with awakenings. Now, we're not sure if that was what's happening with a hot flash or not. Unfortunately, we weren't recording that at the time. But looking at the literature, it was the first time. I've looked at thousands of cortisol records over the years. And I'd never seen anything like this. And then I scoured the literature. No one had ever reported it because nobody studies this, and because it's so difficult. But I have to say, when we had the women in the lab, they were saying it was like the best night of sleep they've had in years. Now, typically when you have someone in the lab, they don't sleep very well. But when we had women in their 40s and 50s come into the lab, it was the first guilt-free sleep they've had in forever. Because they weren't on vacation and now feeling guilty that they're on vacation. And they weren't at home, which means they got stuff to do. And so they were just in the lab. Like, I can't be bothered because I'm in the lab. I'm doing science. And then they felt they could sleep in and they had great nights of sleep.
That's pretty amazing. Well, I would be happy to help you spearhead a marketing plan. I'd ask SCCR if I could be a part of that if you want to. Just let us know.
Very good.
Thank you. I have a question myself. You alluded about the ADHD and misdiagnosis of that because of sleep apnea or sleep deprivation.
Yes.
So what is like, how can we go about making that a more known knowledge out there? Because it seems there are really extreme increase in, prevalence of ADHD diagnosis.
Yes.
And how can we know if a child is having enough sleep? Is it the length of the sleep, or is it the quality of the sleep that defines that?
Yeah, so in a lot of kids, so you actually, so first you did mention sleep apnea, and in some kids, actually sleep apnea causes these same symptoms.
Mm-hmm.
Sleep apnea is just less common in kids, but it can definitely happen. And how to get the information out there? I mean, that's a great question. I think it's incredibly important. We do a lot of work with adolescents, and they're just not getting enough sleep. I mean, if you just look at the quantity, forgetting about the quality, if you look at the quantity of sleep, it's insufficient. And the easy way to tell is that, do they sleep in on the weekends? If they are, then they're not getting enough sleep. If they're not sleeping in, then they're probably okay. But most teens, given the opportunity to sleep in on the weekend, will do so. Now, again, it doesn't lead in everyone, obviously, to these ADHD symptoms. But that's the question. So, for example, if someone has these kinds of symptoms, the question is, well, the first thing to do is see if you can increase the amount of sleep. Like, if they can get more sleep, does that help their symptoms? So before giving them a medication, that's how I would go. But frankly, it's a lot easier to give someone a pill. And so I think, sadly, that is often the default, because it's just much easier to do that than to try to figure out how do you convince a teen to try to get more sleep. That's, I can tell you, a very tricky thing to do. We're working on that. We've got a combination intervention where we do a behavioral intervention that gives them like 10 minutes of sleep, with a passive light intervention where we're shifting their brains to Denver while they're living in Palo Alto. And so they get sleepy an hour earlier. And so we can get them 45 minutes of extra sleep per night that way, when we combine that. So there are ways to do that.
How do you shift them to Denver's time?
We put in a specialized light in their bedroom that has these brief flashes of light, and it does it, and it basically is kind of giving them a fake sunrise earlier than when they actually normally get light, and so it moves their circadian system to an earlier hour, and so their whole sleep system then gets shifted an hour earlier. And then if you convince them to try to go to sleep an hour earlier, now they can, and then they get 45 minutes of extra sleep per night, and that's something that. So we published on that, and we're now testing to see if that lasts. Does that last? Like, we can do it for 6 weeks, but how about for a year? So that's what we're kind of seeing.
That's very interesting. Thank you. Appreciate it. Any other questions? We are at the hour, so I really appreciate it. It was very informative. Thank you so much for your time. And again, for everyone, I will send out a copy of the slides along with a link to our website where the recording will be available in a couple of days. And please, I will put the evaluation link in the chats as well. And please make sure to complete that. And I will also email it to you for the course evaluation along with the slides. So again, thanks everyone for attending. And thank you so much, Dr. Zeitzer, for this informative lecture. Have a lovely rest of the day, everyone. Thank you. Thanks. Bye.